Section 15.2 is about eukaryotic gene expression. In a typical human cell, only about 20% of genes are expressed at any given time. Different cell types, which have identical genomes, turn on different genes to carry out specific functions, meaning that identical genomes, every nucleus has identical 23 pairs of chromosomes. And the differences between the cell types is due to differential gene expression. No, I haven't seen them. So what this means is with the unique cell types like muscle, nerve, skin cells, again, they have identical DNA inside the nucleus, but they need to carry out transcription and translation differently. For example, you wouldn't need lactase necessarily made like in the digestive system with those stomach cells in the nerve cells or in brain cells. So every cell, although having identical genome, they need to carry out protein synthesis differently depending on their cell type. That's what differential gene expression is. This diagram summarizes 15.2 and how eukaryotic gene expression is regulated at different stages. The different stages are indicated by the boxes with the name of the stage in it. For example, at the top, chromatin modification, DNA unpacking. That's going to be one control point where gene expression can be turned on, off, where it can be accelerated or slowed down depending on what the cell needs. And then transcription, RNA processing, transport to the cytoplasm, translation, protein processing, transport to the cell destination. And then off to the left, I missed two, degradation of mRNA and degradation of a protein as well. The first stage where gene expression can be controlled is at the chromatin. So we need a little bit of background again about what chromatin is. So chromatin is tightly bound DNA. It's tightly bound by proteins known as histones. And sometimes you'll see mentioned in books nucleosomes. So as a reminder, what exactly is a nucleosome? Nucleosome is 8 histones tightly bound by DNA. So the tighter the histone and the DNA is wrapped around one another, or the nucleosome is tightly bound, then that means that transcription is less likely, because that process is less accessible when you have the DNA really compacted, really tight. Another way that chromatin can be modified is by DNA methylation. So we need to remember a little bit about our functional groups that we learned a billion chapters ago, I think chapter 2, chapter 3. A methyl group is a carbon with 3 hydrogens, so it's a CH3. So when CH3 is added to the DNA, there is a decrease in transcription because the DNA ultimately gets tightly packed even more. And where the, I did want to note where the methyl group gets added, it typically gets added to cytosine in the DNA molecule, being one of the nitrogenous bases. Another way that chromatin can be modified, again, one of the first control points, is histone acetylation. So this is where an acetyl group is added to the histone, the protein itself. Now, what is an acetyl group? An acetyl group is a carbon that is double bonded to an oxygen. And that carbon is also attached to a CH3. It's also attached to a methyl group as well. So again, acetyl is C double bond to oxygen and also bound to a methyl group. Now, when this is added, because it's being added to the protein itself on the N-terminus, there's an increase in transcription, which is different than the methylation to the actual DNA molecule. So these are two different ways that chromatin modification, one of the first control points of gene expression. You may want to look back if you have your books with you, which I don't know that I would over Christmas vacation, but you might want to review, I think it's chapter 13, figure 1323. That will give you a really good visual of what a nucleosome is and what the histones look like all bound together. Something I wanted to note about the previous slide, that the DNA methylation and histone acetylation, it does not change the actual DNA sequence. So those nitrogenous bases, the A's and the T's and the G's and the C's, those are not altered. Again, when those are altered, that's called a mutation, and that's not what we're talking about here. So we learned a little bit before Christmas about epigenetic inheritance, and these are essentially modifications on the chromatin on itself that can be passed on to future generations. Again, not altering that DNA sequence. So unlike DNA mutations, these changes to chromatin, they can be reversed. So demethylation of DNA is possible, and this often explains the differences between identical twins. As we read about the Holocaust survivors and their children, the methylation often, because it's passed on, we may see it in future generations. And with the identical twins, some examples, the methylation itself, gene silencing, histone acetylation, which I just covered, X chromosome inactivation, heterochromatin, and what I should have mentioned this earlier, heterochromatin is highly condensed chromatin. That's what heterochromatin is. And again, when you have highly condensed chromatin, it can't be transcripted. Transcription can't take place. So that's known as silent chromatin because it's too condensed for that process of protein synthesis to occur. The link at the bottom is a quick 2-minute video going over the epigenome, so you might want to check it out and pause here and jot it down. The first step of gene expression being regulated was with chromatin modification. Depending on how that chromatin and DNA was packed together, regulates whether or not transcription is more or less likely to occur. The second control point of gene expression being regulated is at transcription. So to remind you what transcription is, it is the process of DNA getting read and RNA being synthesized. And in a eukaryotic cell, it's going to be pre-mRNA, like a rough draft version of RNA first. So what exactly is transcription initiation? The second step, again, of regulating gene expression. So this is where we have specific transcription factors that are going to bind to control elements. So there's a couple words that you need to know here. So a transcription factor is a set of proteins that are going to either promote transcription or they are going to decrease the likelihood of transcription to take place. Those, again, are transcription factors. Control elements, these are going to be pieces or segments of DNA that are non-coding. So specific nucleotide sequences where proteins are actually going to bind to it. And that's going to serve a certain purpose in a few minutes on a different slide. So the enhancer region is the area upstream of the promoter region in DNA. So again, you have a DNA strand. What comes first is an enhancer region. Then you're essentially going to have a promoter region. That's where that Tata box is. And then transcription is going to start. Again, transcription being the DNA getting read and RNA polymerase building mRNA. This diagram summarizes transcription really nicely. I would pause and study this diagram. So the strand at the top in blue is going to be DNA, the template strand for transcription. On this strand, you can see two yellow pieces off to the left. These are known as control elements. So what are control elements? Non-coding pieces of DNA in which transcription factors or proteins are going to bind. Now, these transcription factors or proteins are either going to activate transcription, which means it's going to increase the likelihood that transcription is going to take place, or it's going to repress, which means the likelihood of transcription is less likely, or there's a decrease in transcription, depending on, again, these proteins that bind to the non-coding pieces of DNA, known as control elements. Now, the region or stretch of DNA right after that is known as a promoter region, which we already learned. In this region is where the Tata box is and where just after transcription actually starts, which means RNA polymerase is going to move downstream the DNA strand and produce RNA, pre-mRNA to be specific, which we see in the middle line in a pinkish salmon color. So this is the rough draft version of RNA. So please remember that introns get spliced out and removed, and they stay in the nucleus and they get degraded and reused or recycled, but they do not leave the nucleus. The exons get put together to form a final version of mRNA. Once we have all the exons put together, a 5' cap is going to be added, and then a poly A tail is going to also be added. Now, the 5' cap is a modified version of guanine, and the poly A tail is a series of adenine molecules. Also, if you look at the bottom strand being the final version of messenger RNA, you can see this whole 5' UTR. So what that is untranslated region of mRNA. And you also see that at the 3' end as well. So again, please study this. This can be really helpful in summarizing transcription with a little bit new information up at the top with the control elements, transcription factors, etc.